We're going to now consider the basic structure of prokaryotic cells. When we look at prokaryotic cells, obviously they are cells that do not have a membrane-bound nucleus or membrane-bound organelles. This makes them typically smaller than eukaryotic cells. Also, usually means that they're a little bit, for lack of a better way of putting it, simpler than eukaryotic cells. But that doesn't mean that they are simple. You'll see there are surface layers to the cell envelope. There's a cytoplasmic or cell membrane, which is similar to most cell membranes, honestly. The unique cell walls, which we'll discuss, and a capsule may be present. The cytoplasm within the cell. The genetic information of that cell is housed within the nucleoid. This is not a membrane-bound nucleus, obviously, but it's just an area with that genetic information containing the chromosomes of that cell. And then any appendages, such as flagella, amongst others, that may be used for motility. This image shows you the basic structure of a prokaryotic cell. You can see the DNA housed within the nucleoid, organized into various chromosomes. A singular flagellum here. You've got that cell wall or cell envelope, which we'll talk about shortly. The capsule is depicted here in gray, the cell wall in blue, the cell membrane or cytoplasmic membrane here in the bronze color. Multiple ribosomes. You may be thinking ribosomes are organelles. Well, the key is they're not membrane bound, so they're okay for prokaryotic cells. And then you've got a pilus or multiple pili along the surface. Table 3.3, which continues over the next couple of slides, actually tries to detail for you the various functions of some of the structures you just heard me mention. I would tell you to definitely take a look at this table. I will expect you to know those functions related to the various prokaryotic cell structures, but we'll pull those out as we go through the discussion, so I'm not going to reread this table to you. It's the second part to that table. And so we have a quick slide here regarding the cell membrane of a prokaryotic cell. Like I said, very similar to that of a eukaryotic cell. You'll have a phospholipid bilayer with membrane proteins potentially. You've got those hydrophilic phosphate heads as well as the hydrophobic tails composing that bilayer. The proteins, as they do with most cell membranes, can be channels or gates, serve for cell signaling, also can possibly be enzymes. It does follow that same fluid mosaic model that you heard about in general biology, meaning that the components within that cell membrane can shift and move back and forth. They don't flip-flop from one face to another, but they can kind of just drift, so to speak, throughout that surface. Keep in mind that that cell membrane is also going to be selectively permeable, once again sharing a similar characteristic to that of the eukaryotic cell membranes that, as I'm sure you've discussed before in general biology. The bacteria and archaea are going to have similar cell membranes, the major difference being since archaea are typically structured to survive in harsh environments, extreme environments, as we've talked about previously, you may have slightly different phospholipid compositions which impart greater resistance to those archaea since they live in those extreme environments. The cell wall of these prokaryotic cells, we've already mentioned in a previous video. We use that in order to try and accomplish our gram stain, and we can distinguish gram-negative versus gram-positive bacteria. So it's important that we evaluate the structure of the cell wall of these prokaryotic cells. Once again, we have another table for comparison's sake. I would encourage you to go back and take a look at this. You can see the color of each category. We've already mentioned purple for gram-positive cells, pink or red for gram-negative cells. We'll discuss many of the other attributes that you see throughout the table, but I would highlight the peptidoglycan thickness, which is in this table, the presence or absence of the teichoic acids, outer membrane, lipopolysaccharides, and porin proteins as well. You'll see certain cell walls have them, while the other may not. And also the sensitivity to penicillin and lysosine. This is also a characteristic related to the cell membrane structure. So you'll definitely want to go back and look through this table for that information, but as we go through the remaining slides, it will come about. Now, peptidoglycan is found in bacterial cell walls, and is actually a polysaccharide chain formed from subunits called N-acetylmuramic acid, or NAM, and N-acetylglucosamine, NAG. So a lot of times we say that you have NAG and NAM residues. You see the NAG represented by the purple spheres, the NAM by the blue. You can see that they're linked in a glycan chain, just means it's a long chain of sugars. And then projecting from your NAM residues is typically a tetrapeptide chain. It's a chain of amino acids. And it's these tetrapeptide chains that allow for cross-linking between the NAG and NAM layers of the bacterial cell walls. If you notice in the bottom image, you can see layer after layer after layer of NAG and NAM. The tetrapeptide chains used for cross-linking between the layers so that the layers associate with one another, essentially helping compose the the bacterial cell wall. The gram-positive cell wall is essentially a very thick layer of peptidoglycan, somewhere upwards of anywhere from up to 30 layers thick. So you can see the alternating peptidoglycan layers, the NAG and NAM residues, once again the purple and blue spheres. Once again, these will be interconnected by those bridges formed by the tetrapeptide chains. The teichoic acids extend above the peptidoglycan layer, and they're used for a variety of purposes, such as cell signaling and also possibly some receptors. Below the peptidoglycan layer, you have a gel-like material. The key important concept to take away with this gram-positive cell wall is that it's essentially a very thick layer of peptidoglycan. It's analogous to the medieval times when cities built thick walls to protect themselves, so to speak. Essentially, that's what's going on here. A thick layer of peptidoglycan provides the protection when we look at gram-positive cells. By contrast, the gram-negative cell wall is highly different. The gram-negative cell wall begins with an outer membrane. This outer membrane is a bilayer similar to a cell membrane. However, instead of having two layers of phospholipids, instead, the outer leaflet, which is represented here, is made up of a molecule called a lipopolysaccharide, or LPS molecule for short. If you see, it's composed of a sugar, or a saccharide, a lipid, called lipid A, and an O antigen. These lipopolysaccharides have multiple purposes or multiple functions. If you look at it, they can be used as signals for our immune system. The immune system can respond to these lipopolysaccharides when gram-negative bacteria enter the body. Sometimes they function as endotoxins. Endotoxins cause damage to our cells and can ultimately become deadly in certain situations. The lipid A can also be used by the immune system for responsiveness. Then the O antigen. The O antigen is in many cases used to identify specific species. For instance, you may have all heard of E. coli before, but the E. coli can be differentiated based upon their O antigen amongst some other antigens. There's a K antigen associated, for instance, with their capsule, and some other, like I said, other antigens as well. As you see, we're back to the previous image. So you got this outer membrane composed of, at least in part, these LPS molecules. They're on the outer leaflet facing the outside of the cell. The inner leaflet primarily made up of phospholipids. So you see the similarities between the phospholipids here and the inner leaflet of the gram-negative, of the outer membrane, as well as the cell membrane, which is deeper. You'll have proteins, which can serve as channels. These are called porin proteins. They're little pores that will allow materials to move through that outer membrane. Next, you enter what's called the periplasm, or some books refer to this as the periplasmic space. The periplasmic space is found deep to the outer membrane, and this is where you'll find the peptidoglycan associated with the gram-negative cell wall. As you see, it's a lot less than what you had with the gram-positive cell wall. I'll go back to the gram-positive image for comparison. Notice the thickness of the peptidoglycan, and then the relatively less amount which makes this a much thinner layer of peptidoglycan. Deep to that periplasm or periplasmic space, you'll find the cell membrane of that gram-negative cell. The outer membrane blocks the passage of many molecules. That's where the protective aspect of the gram-negative cell wall mainly comes from. It can block the passage of even certain antimicrobial medications or antibiotics. As I mentioned to you before, those porin proteins can allow various items to move back and forth across the outer membrane. The cell membrane, in between the cell membrane and the outer membrane, would be that periplasmic space. It's full of gel-like material, as you heard me mention, in association with the gram-positive cell. And the most important thing that, as I mentioned to you before, is that although you will have peptidoglycan there, it'll be a much thinner layer of peptidoglycan. Now, when you look at microbes, some of them make certain chemicals to try and interfere with the ability of other microbes to grow. Penicillin is an antimicrobial substance actually produced by some molds. These molds, when they produce penicillin, interfere with peptidoglycan synthesis. So, as you saw the two cell walls, the one that was most dependent upon peptidoglycan is the gram-positive cell. Remember, it's a thick layer of peptidoglycan. There's no peptidoglycan in the outer membrane of the gram-negative cell wall. So the gram-positive cell depends heavily on penicillin for protection. If, as penicillin does, you're a medication that interferes with the production of peptidoglycan, you're much more effective against gram-positive bacteria than you would be gram-negative bacteria. Lysozyme breaks the bonds between the linking of the glycan chain, those bonds between the tetrapeptide chains that we saw earlier. So once again, if your cell depends heavily on peptidoglycan for protection, i.e. if you're a gram-positive cell, you're going to have much more effectiveness as if you're using lysozyme than you would as compared to a gram-negative cell. In the end, penicillin and lysozyme are much more effective against gram-positive cells than they are gram-negative cells. We've already talked about the gram stain previously. I won't reinvent the wheel here, so I would refer you back to that previous discussion. But we talked about the fact that the gram-positive cells should appear purple and the gram-negative cells should appear pink. The major reason being due to the difference in peptidoglycan synthesis. The thought or the hypothesis is that as you use the decolorizer, what happens is that the peptidoglycan associated with gram-positive cells constricts and contracts. Notice the thickness of the peptidoglycan here before decolorization, and notice how it becomes more compact after decolorization. That decolorization step, constriction, of the peptidoglycan resists the removal of the iodine crystal violet complexes, and this is why those gram-positive cells remain purple. But the relatively thin nature, as we come down to the gram-negative cell wall, the relatively thin peptidoglycan is affected tremendously by the ethanol we use during decolorization. Notice how the ethanol breaks that peptidoglycan in the gram-negative cell, allowing for a pathway for that crystal violet to move out, and this is why gram-negative cells will be decolorized. Some bacteria actually don't have a cell wall at all. The genus that these bacteria typically belong to is called Mycoplasma. Instead, they have sterols, which you've heard of before if you've taken general biology, because sterols impart strength to a cell wall, to a cell membrane, So the Mycoplasma have these flexible cell membranes containing sterols for reinforcement. Since they don't contain any peptidoglycan, these mycoplasma will be unaffected by both penicillin and lysozyme. External to the cell wall, you'll find capsules and slime layers. These are two components, like I said, outside of the cell wall. Typically, the major difference is in their organization. A capsule is very tightly organized and very tightly adherent to the cell. A slime layer would be more loosely organized and loosely attached. The way I usually remember it, I always think about this show I used to watch as a kid called Double Dare, where they use slime. The slime would dump on the people's heads, but the slime was really runny and liquidy. A capsule, more intact, harder, so to speak. Now, these outer layers, both the capsule and slime layer, can be used for association by bacteria. When they associate in certain situations, they form what are called biofilms. The biofilms consist of a community of cells living and working together. They live and work together for protection and to ensure each other's survival, essentially. An example would be a dental plaque. These biofilms, as they develop within the human body, can be very difficult to treat due to the protective nature that typically accompanies their formation. In some cases, you'll see these capsules and slime layers used for multiple reasons, but the three major ones would be protection, attachment, and also enhancement of pathogenicity. Pathogenicity is basically the ability to cause disease. Flagella. These structures are used by bacterial cells for motility. You can see in these images on the right the various flagella. Keep in mind that they're used for motility. They are composed in bacteria of a protein called flagellin. This is different from eukaryotic flagella, which are typically composed of tubulin. The flagella are used during chemotaxis. Chemotaxis refers to a chemical attraction or chemical movement. So receptors in the bacterial cell walls can be used to determine where nutrients may be or toxins. Clockwise rotation would produce a run in which that organism goes in a straight line. Counterclockwise rotation of the flagella would produce a tumble. The tumbles are random movements meant to try to either elude a toxin or get closer, potentially, to a particular nutrient. Pili are also seen external to the cell wall. They're proteinaceous tube-like structures that are significantly shorter than a flagellum. If you look here on the right, there's a flagellum, much longer. You can see the pili, much shorter. The pili can be used for multiple purposes. If the pili are short and numerous and used for attachment, they're referred to as fimbriae. In some cases, those pili can impart movement. It's a really short kind of jagged movement. It's called gliding motility. So it would take a long time because the organism moves slowly. In other cases, the pili can be used for DNA transfer or conjugation. In that situation, it would be referred to as a sex pilus. Within these prokaryotic cells, you'll find the structure we mentioned earlier in the presentation called the nucleoid. This is where the genetic information, the DNA, will be housed. It's organized into chromosomes. Plasmids are extrachromal pieces of genetic information or DNA. They don't usually provide anything essential to that organism's genetic code. In other words, they can't produce or lead to the production of another organism, but what they usually impart is some sort of distinctive advantage to that organism. It could be that that plasmid codes for a sex pilus that allows it to share its genetic information, or it could be that it provides resistance to a particular antibiotic. The prokaryotic ribosomes are unique as well. They're actually called 70S ribosomes. They consist of two subunits, a 50S and also a 30S subunit. You don't have to pay attention to the math because it's not a direct correlation, but putting together 50 and 30S subunits gives you a total 70S ribosome. The S is for Svedberg unit, which is somewhat of a measurement of their mass, so to speak. So the 70S ribosomes are only in prokaryotic cells, and by contrast, eukaryotic ribosomes are actually 80S. As it says here, this is medically important because medications are designed to target 70S ribosomes, and that means they affect only prokaryotic or bacterial ribosomes, while they will leave eukaryotic 80S ribosomes alone, which means that they would do harm to a bacterium while not harming a human being. Other internal components of prokaryotic cells, they do have a cytoskeleton. That cytoskeleton is proteinaceous in nature, similar to a eukaryotic cytoskeleton. Storage granules usually are used when you have an abundance of nutrients, so that if there should ever be a time when there is a lack of them, there are some on hand. So these storage granules can store anything like carbon or even molecules used for energy. And then gas vesicles are used by some microbes to control their buoyancy. Buoyancy is their ability to float. So we see these quite commonly in prokaryotic cells that are aqueous or that live in a watery environment. Next up, we consider the endospores. The endospores are a dormant cell type typically produced under harsh conditions. Those conditions would be things like extreme temperature, like heat or freezing, chemicals that might try to disrupt that cell, chemical desiccants, or even other types of toxins like bleach, for instance, UV radiation, and even hot boiling water. All of these would be counterproductive for the organisms to survive. Now, there's two genera that typically produce these endospores. Those would be Bacilli and Clostridium species. Those endospores are dormant, which means they're not metabolically active. However, if conditions change, they can become metabolically active. And then we would say they begin to germinate. So germinate implies that the endospore goes from being dormant to vegetative. Vegetative just means that it's a cell that's actively accomplishing metabolic processes. And these endospores are found virtually everywhere. The process of forming endospores is called sporulation. And it's typically triggered by a limited amount of carbon or nitrogen. Limiting these nutrients would deprive the organism of the ability to produce energy. As I've talked to you about before, germination is when that endospore goes from being dormant to being active again. You can see it's triggered by heat or chemical exposure. When we say chemical exposure, we're usually talking about nutrients used by that organism. Key concept to keep in mind, this is not a means of reproduction. It's essentially a means of survival. So the organism doesn't reproduce by forming endospores. It survives that harsh environment and then can become vegetative at a later point. This diagram walks through the steps in sporulation. You can see during sporulation, the cell must first become starved. Once it's starved, the cell can actually start to duplicate its DNA. Once the DNA is duplicated, a septum is formed. And this septum divides the cell. As you notice, the area to the right is a little bit bigger than the area to the left. This is the beginning of the spore formation. The larger compartment begins to engulf the smaller one. And the components within the smaller compartment ultimately begin to form the endospore. The smaller compartment next develops into what's called a forespore. It has a special unique covering, primarily composed of additional layers of peptidoglycan, and this ultimately reinforces the outer lining of that endospore, allowing it to be more resistant to those outside influences. The mother cell is going to be degraded due to the lack of nutrients. Ultimately, it dies, but the spore, along with its spore coat, are still going to be there, and ultimately the genetic information is preserved so that if the environment becomes favorable for growth again, that spore can ultimately germinate and become vegetative. As you see here, the endospore layers, as I mentioned to you before, prevent damage to that genetic information. Highly resistant to heat, chemicals, and other outside influences. The genetic information is very tightly packed. As you can see, it's dehydrated. The lack of water actually prevents heat from damaging it. The proteins that bind tightly that DNA and protect it. That's the whole purpose of the endospore. And then calcium dipicolinate. That's a very important chemical seen within these endospores that helps or imparts more resistance to them. With that, we'll conclude the discussion about the prokaryotic cell structure.